Hello everyone, I am Ms.S.V.Saranya, Assistant Professor, Department of Biochemistry, K.S.Rangasamy College of Arts and Science, Autonomous, Tiruchengode. Today we are going to see a topic about central dogma of molecular biology. The most important task for the cell is to synthesize protein based on codes in DNA. But how these codes are read? What's the process that helps to code the languages? The protein synthesis by the sequence of adenine, guanine, thymine, cytosine. Answer for all the questions lies in the central dogma of molecular biology. What exactly the central dogma? DNA is a genetic material which carries all the information within the nucleus. Replication is a process, and transcription and translation. These are all the step-by-step process which are involved in this central dogma of molecular biology. Replication, transcription creates RNA from the DNA process. Translation creates a protein from the RNA. It was first proposed in 1958 by the Francis Crick, discovered the structure of DNA. Diagram shows that DNA is replicated and it will be transcribed into RNA, and RNA is translated into protein. Let's see the description about DNA. DNA is stored information inside the nucleus. It copied DNA by the replication process. RNA is copied from a piece of DNA to make a single protein, which is transcription process. Then translation process. Translation process is an information from a single RNA used to make a single protein by the translation process. The functions in central dogma. First we are going to see about replication. What is replication? The biological process of producing two identical replicas of DNA from one original DNA molecules. It will be done in three processes, which are all conservative, semi-conservative and dispersive method. Replication process. Replication of DNA are described in three stages. One is initiation, next one is elongation, third one is termination. First, initiation. The process is initiated particularly in an origin, which are targeted by a initiated protein. Sequences used by initiator protein tend to be AT-rich sequences, easier to trans-separate. These inhibitor recruit other proteins and form a pre-replication complex, which unwinds the double-stranded DNA. Elongation process in replication. DNA polymerase add DNA nucleotide at the end of the 3' of the newly synthesized polynucleotide strand. This process will continue until the DNA polymerase reaches the end of the template strand. Primase inhibitor polynucleotide synthesis, and by creating a short RNA polynucleotide strand complementary to template DNA strand called the primer. Then termination. Termination is the final stage of the replication process. A different type of DNA polymerase removes the RNA primer and replace it with the DNA. DNA ligase join two Okazaki fragments with polyester bond to produce a continuous chain. Each new DNA molecule is rewound by helicase. Each molecule is identical. What are all the enzymes which are involved in replication process are given here, which is helicase, DNA polymerase, ligase, primase and DNA gyrase or topoisomerase. First one is helicase. Helicase helps to open up the double strand in the DNA. DNA polymerase, responsible for the actual synthesis of DNA, brings the nucleotides. Next, ligase. Ligase helps to join together small newly synthesized pieces of DNA called Okazaki fragments. Next, primase. It added RNA primer so that DNA synthesis can begin. DNA gyrase. It reveals train of unwinding by DNA helicase. This is a specific type of topoisomerase. Transcription. First process is replication, another one is transcription. Transcription is the first step of gene expression. In transcription, a particular segment of DNA is copied into an RNA. It is done in two waves. During transcription, a DNA sequence is read by an RNA polymerase which produces an complementary anti-parallel RNA strand called the primary transcript. As like replication, transcription process are also having three types in steps by initiation, elongation, termination. Initiation process attachment of RNA polymerase to the promoter region on DNA. Then elongation process. Elongation process is building on the mRNA from the end of 3' end of the DNA. Then termination. In termination, release of RNA polymerase and mRNA following transcription of the terminator region of DNA. These are all the enzymes which are all involved in transcription process. RNA polymerase, histone acetylase transferase, RNA splicing. RNA polymerase holoenzyme signifies that it is a complex of RNA polymerase and multiple other enzymes. Next, histone acetylase transferase to remove the heterochromatinization of DNA segment in the area of transcription. RNA splicing. There are more proteins in any organism than the number of genes present in the genome of the organism. Then translation. Final stage is translation. Translation is the process by which protein are synthesized from mRNA. The amino acids added sequentially in the specific number of sequence. What are all the materials needed for translation, which are all given here? Amino acids, DNA, RNA, enzymes, that is peptide synthetase, amino acetyl RNA synthetase, coenzyme and cofactors. Steps in translation. Translation process follows these steps. Activation of amino acid, initiation, elongation and termination. Activation of amino acids are 20 amino acids are activated before start of protein synthesis. There is one or sometimes two enzymes to activate one amino acid. Then initiation. The process starts with forming 30th subunit and activation that. An initiation complex is formed at 30th subunit of ribosome. Then 70th complex is formed and activated. Then elongation process. In the last stage is elongation process. The finished polypeptide is released to go and do its job in the cell. Central dogma of molecular biology states that DNA containing some instruction which is followed and it will be converted into RNA by the transcription process. Then it will be translated into a functional protein. RNA then uses the instruction to make a functional protein. Thank you.